we are going to explore something we experience every day, yet often take for granted. That's light and how we perceive color. Let's begin with a simple question. What is light? Light is a form of electromagnetic radiation. It is a wave that carries energy through space. It travels incredibly fast, about 300 million meters per second in a vacuum. But not all light is visible to the human eyes. The visible spectrum, which our eyes can detect, makes up only a small portion of the electromagnetic spectrum. It ranges from roughly 400 nanometers, which is violet, to 700 nanometers, which is red. Each wavelength within that range corresponds to a different color. Shorter wavelengths appear violet or blue, while longer wavelengths appear orange or red. When all the visible wavelengths combine, they form white light, the kind of light emitted by the sun or a standard light bulb. Now, when light strikes an object, one of three things can happen: absorption, transmission, or reflection. If an object absorbs all visible wavelengths, it appears black, because no light is reflected to our eyes. If it reflects all wavelengths, it appears white. Most objects, however, reflect and absorb certain wavelengths selectively, and that's what gives them color. For example, when white sunlight hits a red apple, the pigments in the apple's skin absorb most of the wavelengths except those corresponding to red light, which are reflected. That reflected red light enters our eyes, stimulating receptors in our retinas, and our brain interprets the signal as the color red. Color begins with light, specifically visible light, which is just a small portion of a much larger range of energy called the electromagnetic spectrum. If you look at the diagram on the slide, you can see the full electromagnetic spectrum laid out from left to right. On the far left are gamma rays and X-rays. They have extremely short wavelengths and very high energy. Moving to the right, we pass through ultraviolet light, then reach the narrow band of visible light, and beyond that are infrared, microwaves, and radio waves, which have progressively longer wavelengths and lower energy. So visible light lies right in the middle, a tiny window in the spectrum, but it's the only region our eyes can detect. The visible spectrum spans roughly 380 to 750 nanometers in wavelength. The shorter wavelengths around 380 nanometers corresponds to violet and blue light, which carry more energy. The longer wavelengths near 750 nanometers corresponds to red light, which has less energy. Every color we see, violet, blue, green, yellow, orange, and red, falls somewhere within this narrow range of wavelengths. You can think of white light, like sunlight, as a mixture of all these wavelengths combined. When white light passes through a prism, as shown at the bottom of the slide, it bends or refracts, and the different wavelengths spread out to form a spectrum of colors, just like a rainbow. This dispersion happens because each wavelength of light bends at a slightly different angle when it moves from one medium to another. Now, this visible band is incredibly small compared to the entire electromagnetic spectrum. Notice how much larger the regions of infrared, microwave, and radio waves are compared to the narrow strip of visible light. Yet, it's within this tiny range that all of our visual experiences, every color we perceive, are contained. So, to summarize, visible light is a type of electromagnetic radiation, just like UV or infrared. It's the only portion of the spectrum that can be detected by the human eyes. And it spans wavelengths from approximately 380 to 750 nanometers. In the next part, we will discuss how this light interacts with matter, how some wavelengths are absorbed and others reflected to create the colors we actually see in objects. The color that we see from any object, whether it's a red apple, a green leaf, or a glass of orange juice, depends on how that object interacts with white light. White light, like sunlight or light from a bulb, is made up of all the colors of the visible spectrum, from violet through blue, green, yellow, and red. When this white light strikes an object, the surface of that object can do one of three things to the incoming light. It can absorb, transmit, or reflect it. The particular mix of wavelengths that get reflected or transmitted is what determines the color that our eyes perceive. Let's look at the apple on the slide. When white light hits the surface of a red apple, the apple's pigments absorb most of the shorter wavelengths, that is, the green, blue, and violet parts of the spectrum. The longer wavelengths of red, however, are reflected more strongly. That reflected red light enters our eyes, and our brain interprets the apple as being red. So the apple is not inherently red. It simply reflects red light and absorbs the others. Now look at the orange juice on the slide. Here the situation is similar, but the juice interacts with light a bit differently because it's somewhat transparent. When light passes through the juice, the liquid absorbs much of the red, blue, and violet wavelengths, allowing mainly the yellow and orange wavelengths to be transmitted. Those transmitted wavelengths reach our eyes, and that's why the juice appears yellowish-orange. This principle, called selective observance, is what explains the wide variety of colors we observe in nature and in materials. Different substances have molecules or pigments that absorb certain wavelengths more strongly than others. The light that remains, whether reflected or transmitted, is what defines the object's apparent color. So, in summary, white light contains all colors. Objects absorb some wavelengths and reflect or transmit others. The color we perceive corresponds to the wavelengths that reach our eyes, the ones that are not absorbed by the material. In essence, the colors we see are not properties of the objects themselves, but rather the result of how the object interacts with light and how our eyes and brain interpret that reflected or transmitted light. Now, let's take a closer look at the absorption spectrum of a potassium chromate solution, which has a distinct yellow color. On the graph, the x-axis represents the wavelengths of light in nanometers, ranging from around 380 to 680 nanometers. This covers the visible spectrum from violet through red. The y-axis shows the absorption, which tells us how much light is absorbed at each wavelength. If we examine the spectrum carefully, we can see that the potassium chromate solution strongly absorbs light in the violet and blue regions, roughly between 380 and 450 nanometers. This is shown by the tall peak in that area. The absorption that drops significantly as we move toward the green, yellow, and red regions of the spectrum, meaning that the solution does not absorb much light at those wavelengths. Now, remember that the color we see in a solution comes from the light that is transmitted or reflected, not the light that is absorbed. In this case, because potassium chromate absorbs violet and blue light, the remaining colors, primarily yellow and red wavelengths, pass through the solution and reach our eyes. The combination of these transmitted wavelengths gives the solution its yellow appearance. So, in essence, the yellow color of the potassium chromate solution is a result of selective absorption. It absorbs the high-energy violet and blue light and transmits the lower-energy yellow to red light. This concept is a cornerstone in spectrophotometry because by analyzing which wavelengths are absorbed, we can understand not only the color of a substance, but also its electronic structure and chemical composition. The absorption spectrum acts like a fingerprint. It tells us how a substance interacts with light, which is vital for identifying compounds and studying their behavior in solution. Now, let's explore how we actually perceive color, that is, how our eyes and brain interpret the colors of substances based on the light they absorb and transmit or reflect. White light is a mixture of all visible wavelengths. When the white light shines on a substance, that substance will selectively absorb certain wavelengths and transmit or reflect others. The color that we see is not the light that's absorbed, but rather the light that remains and reaches our eyes. To better understand this, we can use the color wheel shown here on the slide. The color wheel helps us visualize the relationship between absorbed colors and observed or transmitted colors. For instance, if a solution absorbs light in the red and orange regions of the spectrum, those colors are being removed from white light. What remains, primarily blue light, reaches our eyes, so we perceive the solution as blue. Conversely, if a substance absorbs blue or violet light, the remaining colors, mainly yellow or orange, are transmitted or reflected, making the solution appear yellow or orange. You can think of this in terms of complementary colors. The color that appears is opposite to the color that is absorbed on the wheel. If a substance absorbs violet, it looks yellow. If it absorbs red, it looks blue-green. If it absorbs green, it appears magenta or red-violet. This principle is exactly what we saw earlier with potassium chromate. Its solution appears yellow because it absorbs violet and blue light while transmitting yellow and red. Light is a type of wave called electromagnetic radiation. Let's find out what a wave is. A wave is defined as a disturbance that travels through a medium, transferring energy from one particle of the medium to another without causing any permanent displacement of the medium itself. That means, while the wave moves forward, the individual particles of the medium simply oscillate back and forth without causing any permanent displacement of the medium itself. For example, when you throw a stone into a pond, the ripples that move outward are waves. The water itself doesn't travel outward. It just moves up and down as the energy of the disturbance passes through. Let's identify some key characteristics of waves. Wavelength is denoted by lambda, and it is the distance between corresponding points on two adjacent waves, such as from crest to crest or trough to trough. It tells us how long one complete cycle of the wave is. Amplitude is denoted by capital A. Amplitude represents the height of the wave, measured from the midpoint to the crest or trough. It indicates the intensity of the wave. Frequency is denoted by nu. Frequency is the number of waves that pass a given point per unit time. It is usually measured in hertz, which means cycles per second. A higher frequency means more waves are passing each second. Wavelength and frequency are inversely related. As one increases, the other decreases. This relationship is expressed like this. Nu is directly proportional to 1 over lambda, or nu is inversely proportional to lambda. This means that shorter wavelengths correspond to higher frequencies, and longer wavelengths correspond to lower frequencies. For instance, gamma rays have extremely short wavelengths and very high frequencies, while radio waves have very long wavelengths and very low frequencies. This slide presents the electromagnetic radiation, a continuous range of electromagnetic waves characterized by their wavelengths and frequencies. On the horizontal axis, we see wavelengths measured in meters, ranging from incredibly short gamma rays at around 10 to the negative 12th meters to long radio waves stretching up to 10 to the 3rd meters, or 1,000 meters. Just below that, we have the frequency scale measured in hertz, which runs inversely to wavelength, from high frequencies around 10 to the 18th hertz for gamma rays down to low frequencies near 10 to the 4th hertz for radio waves. The leftmost wave has a high frequency and short wavelength, characteristic of gamma rays or X-rays. The middle wave represents a moderate frequency and wavelength, typical of visible light. The rightmost wave has a low frequency and long wavelengths, like radio waves. Zooming into the center of the slide, we focus on the visible region of the spectrum, the only part detectable by the human eyes. This spans wavelengths from approximately 380 nanometers to 750 nanometers. The color gradient illustrates how different wavelengths correspond to different colors. Violet light has the shortest wavelengths, around 400 nanometers, and highest energy. Red light has the longest wavelengths, around 750 nanometers, and lowest energy. This narrow band is just a tiny slice of the full electromagnetic spectrum, yet it's responsible for all the colors we perceive in daily life. In the previous slide, we learned that wavelengths and frequency are inversely related. It is illustrated in this picture. As wavelength decreases from right to left, frequency increases and color becomes more to violet. These are three waveforms representing electromagnetic waves of different frequencies and wavelengths, illustrating as frequency increases, wavelengths decreases, but energy increases. This inverse relationship is key. As wavelength decreases, frequency and energy increase. This reinforces the principle higher frequency means higher energy, which is why gamma rays are so powerful and potentially harmful, while radio waves are relatively benign. Now, let's try an online activity to reinforce our understanding of the nature of light. Log into the course site on D2L. Click the Content tab at the top. In the left menu, open Lecture Material, then select the module titled 06 Nature of Light. Under the module, find Lab 06 Nature of Light and download a lab worksheet and an Excel spreadsheet. Complete both as you work through the online simulation. Return to D2L and under the same module, open Simulation Nature of Light. In the simulation, select the Beer's Law window. Select nickel chloride from the solution list at the bottom. Make sure that the concentration is set to 350 millimolar by using the slider at the bottom. Turn on the light source on the left by clicking a red button. Drag the meter and place it behind the sample cuvette where the transmitted light passes. Make sure that absorbance is selected. Select Variable in the Wavelengths panel. Open the downloaded Excel spreadsheet and select the worksheet labeled Green NiCl2. Return to the online simulation page and read absorbance values at increasing wavelengths in 5 nanometer increments, starting from 380 nanometers. Record the absorbance values on the Excel sheet. Examine the shape of the plotted spectrum and read the wavelength at the maximum absorbance. Refresh the simulation window by clicking on the Refresh button. Select Drink Mix from the solution list at the bottom. Make sure that the concentration is set to 400 millimolar by using the slider at the bottom. Turn on the light source on the left by clicking a red button. Drag the meter and place it behind the sample cuvette where the transmitted light passes. Make sure that absorbance is selected. Select Variable in the Wavelength panel. Open the downloaded Excel spreadsheet and select the worksheet labeled Drink Mix. Return to the online simulation page and read absorbance values at increasing wavelengths in 5 nanometer increments, starting from 380 nanometers. Record the absorbance values on the Excel sheet. Examine the shape of the plotted spectrum and read the wavelengths at the maximum absorbance. Open the downloaded lab worksheet, follow the step-by-step instructions, and complete Activity 2.